back with our next class. In today's class, we are going to discuss certain various important topics related to osmosis that was discussed in the last class. So let us start today with osmotically active solutions. Okay. Now see, suppose in a clean glass jar, I introduce a solution, say solution A. Okay. And in that solution, I introduce an animal cell. The cell cytoplasm will also act as another solution because cytoplasm is also 90% water after all and 10% dissolved solutes, isn't it? So, say cytoplasm acts here as another solution B. Alright. And in between the two solutions, we have a plasma membrane which will act as the semi-permeable membrane, right? So every apparatus required for osmosis to take place is present now, right? Now, say here, solution A has lower solute, whereas solution B has higher solute in comparison to solution A. So now, as per the definition of osmosis, you very well know that water moves from a region of lower solute to higher solute, isn't it? So, water would start entering the cell via endoosmosis. And in no time, you will find that the animal cell will swell up and eventually burst. So, such a solution in which the solute concentration is lower in comparison to the cell cytoplasm, where an animal cell would eventually burst, would be known as hypotonic solution. Alright. This is one of the osmotically active solution. Now, second, say this time I take into consideration the same apparatus. I fill a glass jar with solution A, alright, and then introduce in it an animal cell, the solution B, alright. But this time solution A has higher solute and B, that is the cell cytoplasm, has lower solute in comparison. This time what will happen? Water would start moving from lower to higher solute. That means from the cell to into the jar, isn't it? So that means water will start running out of the cell by exocytosis, right? So in no time the animal cell can be seen to shrink. So it will undergo shrinkage. And this type of a solution where the solute concentration is higher in comparison to cell cytoplasm is known as hypertonic solution. Alright. Now, third and the last osmotically active solution, solution A and solution B, that is the animal cell. This time both A and B have equal solute. So if there would be equal solute in both, so what will happen? The amount of water running into the cell will be equal to amount of water running out of the cell. Hence the net movement of water would be zero and hence the cell will remain as it is without any change. So a particular solution in which the solute concentration remains same in comparison to the cell cytoplasm, where an animal cell will undergo no change, would be referred as isotonic solution. I hope all of you are clear, right? So hypotonic solution, hypertonic solution and isotonic solution. And you know that for an animal cell in hypotonic solution, it would swell and burst. In case of hypertonic, it would undergo shrinkage. And in case of isotonic, there will be no change, right? Now in the same situation, if I introduce a plant cell instead, okay, Same apparatus, but instead of a animal cell, if I introduce a plant cell in these solutions, what would happen? See, in case of a plant cell being introduced in a hypotonic solution, water would start entering inside by endoosmosis even inside the plant cell, because the cell wall of the plant cell is freely permeable, and the plasma membrane, it will act like a semi-permeable membrane here itself, allowing water to pass into the sap vacuole. So as the water starts running into the plant cell, the sap vacuole will start to enlarge and it will start to take in more and more of water. After a while, when the sap vacuole is completely filled up with water, there would be a pressure generated inside the plant cell. This pressure will be from sap vacuole towards cell wall, and we call this pressure to be turgor pressure. Okay, turgor pressure, by which the sap vacuole will start pressurizing the cell wall. Cell wall also being very rigid, it would not burst. Rather, to withstand this pressure, it will exert an equal and opposite pressure towards the sap vacuole. And this pressure would be known as wall pressure. Okay? Turgor pressure and wall pressure both will be equal and opposite, hence they will nullify each other. But because of this reason, what will happen? The cell membrane of the plant cell will remain tightly adhered with the cell wall. Okay? There will be no region in the plant cell where the cell membrane and cell wall would separate. They would rather remain closely, pressed with one another. And in this state, we call the plant cell to be turgid. So whenever you place a plant cell in a hypotonic solution, you will find the plant cell will reach turgidity. Alright. Now, before I go into hypertonic solution, let's go into isotonic solution. What will happen in case a plant cell is introduced to an isotonic solution? If in case a plant cell is introduced to isotonic solution, in that case, the amount of water running inside is equal to amount of water running outside, right? So, will there be any turgor pressure generated? No. And in case there is no turgor pressure, obviously there will be no wall pressure, right? So, in such a situation, the plant cell, it won't be able to retain its turgidity anymore. The cell membrane and cell wall will remain together, but there will be no oppression between them. The adherence between them will be lost. Alright? In such a state, we call the plant cell to be flaccid. Alright? So, in case of an isotonic solution, when a plant cell is introduced there, the plant cell will lose its turgidity and rather it would become flaccid. Fine. Now for the main part, that is what would happen if a plant cell is associated or it is introduced in a hypertonic solution. See, in case the plant cell is introduced in a hypertonic solution, at first, water from the sap vacuole will start to run out. Okay? And eventually, the sap vacuole, due to loss of water, it would start to undergo shrinkage. The sap vacuole will shrink. As it shrinks, it will start pulling the plasma membrane towards the center, right? This will separate the cell membrane from the cell wall, but at the corners. This separation will begin at the corners, alright? Eventually, the whole protoplasm will shrink. The whole protoplasm will shrink and it will kind of gather at the center of the cell. Okay, the whole thing will shrink and will gather at the center of the cell, and this gap areas, they will be filled up with the hypertonic solution itself. Okay, so this particular state when a plant cell is introduced to a hypertonic solution, due to which it undergoes exoosmosis, all the water from the protoplasm is lost and the whole protoplasm undergoes shrinkage. This particular state is known as plasmolysis. So whenever I introduce a plant cell to a hypertonic solution, it will undergo plasmolysis. Is that clear? Now, plasmolysis consists of 3 stages. In the first stage, when plasmolysis just begins, at that time, turgor pressure turns 0. Tp becomes 0. Okay. So, this is the first step of plasmolysis, which is known as limiting plasmolysis. Alright. Second step is when the water has considerably run out of the sap vacuole, due to which the cell wall and cell membrane have started to separate at the corner. This is the second step when Tp becomes negative, and we call it incipient plasmolysis. And finally, when the entire water has run out, and this is the last step when the whole protoplasm have undergone shrinkage and it has gathered at the center of the cell. This is the last step called evident plasmolysis. Here Tp further becomes more negative. Alright, so understood this? Fine. Do remember, plasmolysis is a reversible process. If a plasmolysed cell immediately after plasmolysis is introduced to a hypotonic solution, it can again regain back its original state. Alright, it can again gain back its turgidity, and this is known as deplasmolysis. Alright, I hope plasmolysis is clear with you. Next, let's discuss imbibation. So now let us discuss imbibation. Imbibation is basically the absorption of liquid particles on the surface of a solid. Absorption of liquid particles on the surface of a solid when the liquid and solid would remain together but without forming a solution, that would be referred as imbibation. Alright. Now, in imbibation there are, that means, 2 things which are participating. One is the solid particle, that is known as imbibent. Another is the liquid particle, that is called imbibate. Alright, imbibent and imbibate. During imbibation, both imbibent and imbibate undergoes certain kind of changes. They undergo certain kind of changes. Now, let us first discuss what is the change that the solid would undergo. See, whenever liquid particles are adhered on the surface of a solid, the solid undergoes expansion in volume. Its volume expansion would occur. Alright, its volume will expand. It will undergo expansion in its volume. Okay, like let me give you a very common example from your day-to-day life. Like, say in the rainy season, you might find that the doors and the windows which have wooden frames, they might start to expand, or they kind of become difficult to close and open, right? That happens because wood, being an excellent imbibant, it starts to absorb moisture present in the air. Due to the liquid particle or the moisture adhered on the surface, the expansion of volume of the wooden frames of the doors and windows would take place. That's why opening and closing the windows would become difficult then. Alright. Now, next, what would be the change occurring in the imbibate or the liquid? See, in the liquid, whenever the liquid is entrapped on a solid surface, its kinetic energy would change into heat energy. and hence the liquid particles would start releasing heat. And that heat released is called heat of hydration or heat of waiting. Alright, understood? So whenever imbibation would take place, the solid particle would undergo expansion in volume and the liquid particle would undergo it will release heat, that is heat of hydration or heat of waiting. Alright. Now, apart from these two, there is another important thing associated with the imbibation phenomena, that is IP. Imbibation pressure, which was previously denoted as matrix potential, that is psi m. Okay. See, whenever, suppose I take a large piece of rock, and in one of the gaps that is present in the rock, if I fit in a piece of wood being an excellent imbibant, right? So if I fix a piece of wood in one of the cracks present on the surface of the rock, you have to also make sure that the fitting has to be very tight. The wood has to fix in the gap very tightly so that there is no space for the wood to undergo any kind of movement. Okay, so in this state, if you put this rock along with the wooden piece inside water, what would happen? The water would start adhering on the surface of the wood, and hence the wood, being an excellent imbibant, would start to undergo volume expansion. But is there any gap for it to undergo expansion here? No, right? So in such a condition, if you keep the rock for a while, you will find that as the wood is trying to expand in volume and it is not able to do so, that's why a huge pressure of very high magnitude, a pressure of high magnitude will be developed in this portion. That's called imbibation pressure. Imbibation pressure is so high that it can easily break the rock into two pieces. So prehistoric man, using this technique itself, used to break rocks or any such heavy particles. Understood? So imbibation is also associated with a very high magnitude pressure, that's called imbibation pressure. Alright, are you clear about it? Fine. So imbibation is responsible for various important phenomena that happens in your day-to-day life, like seed germination, seedling arising from the soil, etc. Alright, these are important examples of imbibation that you can see all around you. Now, last thing regarding imbibation that you should remember is that among the various plant parts, the best imbibant would be phycocolloids. Not everything can undergo imbibation at an equal ease, right? So the best imbibant among the plant parts would be regarded to be phycocolloids, followed by pectin, protein, starch, and cellulose. Do remember this sequence. It's as per descending order. The best being phycocolloid, followed by pectin, protein, starch, and cellulose. Alright, so this being said regarding imbibation, I hope all of you are clear about it. In the next class, we'll start with long distance transport. As in today, we complete the short distance transport for you for this chapter. Thank you.